We're going to talk a little pharmacology today, and we're going to talk about the pharmacology of alcohol, and you guys are all going to pretend you're alcohol molecules. So, anybody ever hear of the word pharmacology other than our title?
Yes.
Great.
Yeah.
So yes, pharmacologists study how drugs work. And we have basic principles. The major classifications are pharmacokinetics, and as you can see here, the classic aspects of pharmacokinetics deal with how the drugs are absorbed into the body, how they move around in the body, that's the distribution, how they are metabolized or handled by the body and converted into something else, and how they are excreted and removed from the body. And the other side to the pharmacology is what we call pharmacodynamics, or understanding how the drug works. How does the alcohol do what it does? Why does it produce adverse effects? Why does it produce toxic effects? And very important, actually, is what happens to it when there's another drug in the body. How does that change the way alcohol works and how the other drug might work? So we're going to talk a little bit about these things in the context of things that you guys all teach anyway. Okay? So as I mentioned, these concepts for the biologists in the group involve everything from cell types to membranes to membrane transport processes, understanding a little bit of anatomy, the difference between the right and left circulation, understanding some genetics. That's an aspect too that's very important in biology, and we can talk about it in the context of alcohol. What about the chemists in the group? You'll recognize some of these categories, understanding a little bit about polarity of molecules, bond formation, concentration gradients, chemical reactions, and how enzymes function as catalysts. All of these are topics that I know you guys talk about at some point in some of your classes. So keep these in mind. We're going to come back to them at the end. But in between, as I mentioned, we are all going to pretend we are alcohol molecules or ethanol molecules. And let's talk a little bit first about this journey. We're going to take a journey through the body. Okay? So normally we come into the body, most people come in, by drinking alcohol. Everybody would agree with me on that. And coming in to the stomach. Okay? And those alcohol molecules then will go from the stomach into the intestine, and they will be absorbed into the bloodstream, shown here in the little blue venous system leading from the gut to the liver. All right? And if you notice the slide here, it's kind of an interesting slide because it separates the circulatory system between the right and left. But if you notice here, it shows ethanol coming in by inhaling it. Has anyone heard of anything like that? No one's heard about this new device, huh?
Yes. Yes.
Oh, good. Yes, there is a new device out on the market where you can breathe in ethanol and get it right to your brain really fast. So you can get nice and high. And one of the things that we know from research in neuroscience is that the faster a drug gets to the brain in terms of its psychoactive properties, the faster, the more likely it will produce addiction. So yes, this is a very good product to have on the market, and we will see 10 years from now what kind of havoc it's wreaked. Okay, so we're going to come back to this drawing several times. I don't want you to study it just yet, but we're going to. Okay, so if you look at the cartoon here, then you can see that the ethanol is coming into the stomach and it goes into the intestine. Who can tell me what is going to limit the rate at which alcohol moves into the intestine?
It's going to be the concentration of alcohol in the bloodstream and wherever it's going.
But we are alcohol molecules and we are not in the blood yet.
Oh, you're okay. Excuse me.
We're in the stomach.
So it's between the intestinal or stomach wall and the.
Somebody
blood.
said food. Food. Food is the right answer. Because when food is in your stomach, your pyloric sphincter closes, doesn't it? and so you can digest your food. Your gastric acid gets secreted, you digest your food, and then it opens. And then the alcohol can go into your small intestine, where the surface area for absorption is huge compared to your stomach, right? You've got a lot more surface area here for absorption. So most of your alcohol is absorbed through the small intestine into the bloodstream that goes to the liver. Okay? All right, well, let's look and see exactly what happens here. So we ethanol molecules are going to move across the cells that line our gut, the epithelial cells. These are cells that have contact with the external environment, and that's where they get their name, epithelial. Okay? And these cells line your whole GI tract, and they, of course, are lining every single organ in your body that has any external contact, including your skin, epithelial cells. So they're in your bladder, your nasal passages, your gut, etc. And you can see by the arrows that they're moving with the concentration gradient. We call this passive transport. No energy is required. And it's pretty easy for these ethanol molecules to move through these cells with passive diffusion or passive transport. And we're going to go through this in a minute. So let's see what happens. They enter the space in between the epithelial cells and the capillary, and that space is called the interstitial space. Okay? Well, let's look exactly at what happens when we pass through the membrane. Now, you guys will all find this as something you recognize, a garden variety lipid bilayer membrane showing the phospholipids lined up with the phosphate head groups on the outside and on the inside of the cell. These are the charged parts of the phospholipids. And of course they're charged because there's water out here on the outside and water on the inside. There's Gail, we see Gail. And basically there are ways in which water can move through these membranes. There are two basic ways water can move through the membranes. You see the proteins sitting here in the membrane? Everybody see that? Well, there are little spaces that the protein makes next to the lipids, and the water can wiggle through. Okay? Now, the water can't go right through the core of the membrane, because that's lipid, and water does not like to mix with fat. But if it finds a little pore or a little space to go through, and if it's big enough, it can squeeze through. Same thing here in this protein. There's a channel right through the middle, and it's a water pore. And water can move through that channel along with solutes that are dissolved in the water, as long as they're small enough. All right? Now, why am I telling you about water? Everybody look at the next slide. Okay. Ethanol is polar like water, and it's a very small molecule. And if you remember what your water molecule looks like, there's your oxygen and your two hydrogens. And everybody knows, of course, that water is a polar molecule. The pole is created by this oxygen that's a very electronegative atom, and it pulls those electrons, tries to pull them away from the hydrogen atoms. It's sort of a tug of war, all right? And this gives the oxygen a little bit of a negative charge, partial negative field. And of course, that leaves the hydrogens with a partial positive field. And this uneven distribution of the charge gives it a pole, okay? And that's why it's called polar. And if you look up here at the ethanol molecule, you see there's an OH group. There it is, the oxygen in red. And that oxygen is doing the same thing in this ethanol molecule as it is in the water, all right? And so it creates a partial negative field around the oxygen, pulling on those hydrogens, pulling on this hydrogen and pulling a little bit from the carbon, okay? And because there are two carbons in the chain of ethanol, it actually gives ethanol a very interesting characteristic. It gives it a little bit of non-polar character, a little bit of lipophilic character, a little bit of lipid-loving character. And alcohol is a very funny molecule. It can dissolve in both water, and it can move through non-polar areas like lipid membranes too. Not all molecules can do this. And it's small. And if you look at other alcohols and increase the number of carbons in the chain of these alcohols, you'll see they go from infinitely soluble in water to more and more and more nonpolar till they finally can't dissolve in any water at all. And of course, that's because there's a greater distance that the charges spread over the carbons, okay? And therefore, you lose the poles. So they're non-polar. All right? Everybody with me? Any questions so far? Okay.
I got one.
Okay, who's asking?
Jim in Texas.
Great.
Tell me what the 9, the 2.7, the 0.6 is.
I think those are the solubility in water. Those are the relative solubilities in water. Sorry, insolubility.
It's grams per 100 grams of water. So, for example, in butanol, 9 grams of butanol will dissolve in 100 grams of water.
Okay.
And let's continue. All right, so here's why I showed you the membrane and the water. Because although alcohol is, or ethanol is polar, it can do something water can't do. If you look carefully at the little cartoon, you'll see that ethanol goes wherever water goes. And that we call that, by the way, filtration, right? Those of you who teach this in biology, it's moving by filtration. It's a passive transport process. There's no energy required. It moves with the concentration gradient by filtration because it's small enough to fit through the little holes. Okay? And just like water, remember? But look in the middle of the membrane. It's also going right through the lipid core of the membrane. And that's because those 2 carbons in that ethanol is just enough to allow it to do that. And that is passive diffusion. Not filtration, that's passive diffusion. Okay, no energy required, moving with the concentration gradient. Okay. What happens to us next? We are in the interstitial space. We got through the epithelial cells by passive diffusion and a little filtration. And now we are going to enter the capillaries. So we're going to go through yet another membrane, okay, in order to get into the bloodstream. And if you look at the capillary in cross-section, here's actually a side view, you can see that the cells of the capillary, and these are called endothelial cells, because they are inside the body. Endo meaning inside. They have no contact with the external environment. So all of your organs in your body, including your blood vessels that are internal organs that have no external access, will be lined with endothelial cells. And you can see that they're kind of loosely packed together. They're not very tight. And if you look at the little blow up here, you'll see 2 endothelial cells come together to make a capillary. You see that? And there's a space in between them, right there. And those spaces, or those pores, they're kind of like pores, they're just loose spaces, basically allow water and solutes to move back and forth. Okay? Actually, pretty big molecules can move through that, and that's because there's such a big space there. All right? Now, these cells also have transient pores that open and close all the time, and these pores are called fenestrae. And anyone that's ever taken Latin, or if you know Spanish or French, you know the word for fenestrae or fenester is window. And it's like little windows, opening and closing, and allowing water and solutes to move in and out. So, you know, relative to the other membranes, the capillary membranes are pretty leaky. And let's see, how about somebody in South Carolina telling me why they're so leaky? Why would we have all these holes?
You'll have interstitial fluid and things leak out of capillaries when you have like a histamine response, like some kind of invasion or a infection of sorts. So you'll have swelling in the area, leaky vessels. Helps to clean the area up as well as,
Yeah.
phagocytize some of the bacteria that come into the area.
Right. Yes, was certainly in response to injury, yes, as part of the cleanup of the mess. But also in terms of our own nutrition. How else would we get large water-soluble molecules from the bloodstream into our tissues? How about something like vitamin C? Big and charged. How would you get that through a biological membrane? You can't use passive diffusion. It's too big and it's big and charged, right? So either a transporter protein pulls it through a membrane, or when it gets to a capillary, it can go through these big holes. Okay? So there's a good reason for that. Oh, this is a great picture. This is a scanning electron micrograph of a capillary, a real one. And it's been colored, of course, to make it look pretty. But if you look really carefully, you can see the two endothelial cells. Here's one. And there's the space in between right there. You see that? And there's the other one. And you can see that the diameter of the capillary is about a little bit bigger than the diameter of a red blood cell. I just think this is a great picture because it shows it so nicely. I did not make that one. Okay, so here we are, us ethanol molecules. We just got into the capillaries. Now what happens to us? Well, here we are, entering the bloodstream. This is the venous circulation. We're on our way to the liver. And we go everywhere in the body. Okay, we're going to enter the venous circulation, going to travel back to the heart with the veins, enter the right side of the heart, where we then get pumped to the lungs. We go with the deoxygenated blood to the lungs. We travel then with the oxygenated blood from the lungs back to the right heart, and then we get pumped out into the arterial system, where we go everywhere to all of our organs, including the brain, which of course, if ethanol didn't get to the brain, we wouldn't drink it, right? I want to hear some rights.
Right. Yes.
Good,
Right.
good.
Right.
Just want to make sure you guys are with me. Okay. And of course, we'll talk a little bit later about how ethanol gets out through the lungs. Not very much of it. Well, let's see. Where exactly does it go in the body? When I said it goes everywhere, it goes to all your tissues, but it does get relatively excluded from something. And actually, alcohol gets excluded from your fat spaces. And it turns out that it goes to different places in the girls and versus the guys. And that has to do with how much fat we have in our bodies. And it turns out that women, unfortunately, have less water space relative to our fat than do the guys. And if you look at the little cartoon here, you can see that for the same drink, the ethanol is going to distribute in the water spaces because it loves water, right? It's polar, in a smaller space. So the sort of classic numbers that were used to describe the percentage of water of our body mass in women back in the early 1900s when these studies were done was about 55% water for women and about 68% for men. Now, these estimates vary wildly, and by today's standards, they've actually probably changed to be more like 49, 50% for women and higher for men, maybe somewhere around 55%. And of course, that is due to the changing of our body compositions over the decades. We have become a fatter society. And so a larger percent of our body weight, our body mass, is fat. So what this does is actually is a wonderful illustration of the concept of concentration based on volumes and percentages. And so if you think about it, this is one of the reasons why women will have a higher concentration of alcohol in their blood, a higher blood alcohol concentration versus a guy for the same drink. Does that make sense to everyone?
Yep.
Good.
Yep.
Now there are some other reasons as well, and we're going to talk about that in a little bit. Now, back to where we are in the body. We're going all over wherever the water spaces are, and of course your bloodstream is one of the water spaces, right? And I did say that it gets into the brain very easily, actually. And it turns out that the brain doesn't like a lot of substances getting into it, especially things that can harm it. And so the brain has a special system called the blood-brain barrier to keep things out. And the blood-brain barrier consists of a little bit of physics and a little bit of chemistry and a little bit of biology. So the physics here, of course, is things are closed up tight. And you see these spaces that were in between the endothelial cells in our garden variety capillary are not there in the brain. They're very tightly bound together by certain proteins. And in addition to that, if you look at this blow up, you can see that there are other cells of the brain that come in. They're called astrocytes. And these cells wrap a layer of fat around the capillary. And this means that only compounds that have lipophilic character to them are able to get out of the blood, out of the capillary, into the brain. And remember, alcohol, although it's polar, has some lipophilic characteristic to it. So it can move, this time now, only by passive diffusion, not filtration, through the membrane into the brain. Okay, there are no little fenestrae here. This is a protective mechanism for the brain. And isn't it interesting that all those drugs that get into the brain that do bad stuff to us are all highly lipophilic? Right?
Right.
Yes. Drugs like nicotine, cocaine, heroin, all that stuff that wreaks havoc does so because they're highly lipophilic. All right, well, here we are in the brain. Here's a little capillary drawn in here, and it's drawn in on top of, actually, these are real neurons that a colleague of mine took a picture of under the microscope. Aren't they pretty?
Yes.
I thought so. The little green molecules here we drew in, and you can pretend that these are the ethanol molecules diffusing out of the capillaries into the space, the interstitial spaces around the neurons. And what do? They basically interact with targets on these neurons to cause the effects that they do, that alcohol does. What does it do? Well, you guys know all this. At the lowest concentrations or doses of alcohol, or with the first drink, of course, sometimes we get a little excited. We may even move into that obnoxious category, right? And then we move into a continuum, everything from poor judgment and impaired memory, poor thinking, into a little bit of balance loss, nausea, vomiting, passing out. We lose our drive to breathe and our cardiovascular drive, and then eventually, of course, coma and death. And so this is the continuum. Of course, the intoxication can be anywhere along here depending on who you are, right? And what your history is in terms of alcohol use. Well, let's look a little bit at what happens. So depending on where in the brain the alcohol is interacting, that will dictate what some of the responses are that were just on that little cartoon. And by the way, I just want to tell you that when you guys start using the curriculum, when we show you the curriculum, all of these pictures are from the curriculum. So you're going to have all these pictures, okay? And many of them are animated. So like that one that I showed with the little beer and the girl and the guy and the percentage, that's animated. It's very, very cute. I think you'll like working with them. All right, what happens? So you know there's a part of the brain, in the midbrain and in the little bit in the front, called the pleasure center or the reward pathway. Some of you may have heard of this. And of course, that is one of the first areas to be affected by alcohol, and it's what makes us feel good when we drink it. It's what's pleasurable about it. And of course, it's also one of the areas that gets activated a lot to make you want to drink another one or a third one. But of course, then the poor judgment part kicks in. Makes you want to go for a fourth or a fifth. And this is part of the frontal cortex. Frontal cortex is very interesting because this part of the brain is the last part of the brain to develop And in the adolescent brain, it is not yet matured. So the parts of the brain that are most important for judgment, for risk-taking behavior, and for impulsive behavior are not yet matured. In fact, it isn't fully matured until about age 24. And that's very interesting why, there's, why do 17-year-olds do what they do? It's because this part of the brain is immature, and then you add on top of it a drink or two, and you can understand exactly the biology of why it happens. It's not just that they're, bad people. It's the biology. Okay? And what else can happen? Well, they can also have memory loss, short-term memory loss, from alcohol acting on a part of the brain called the hippocampus. We'll come back to this in a few minutes. Let's look at some other places. I'll get out of the way here so you can see the effect of the alcohol on, anybody know what this is? I know the biologists do. Cerebellum, yeah. That's what accounts for your coordination and balance. And there are parts of the brain and the brainstem, centers in the brainstem that govern your vomiting, believe it or not. And in fact, this is a very interesting piece here because as the alcohol concentration rises, that vomiting center in your brainstem gets triggered, and you feel nauseous and you may vomit. And the other side to that is there is a problem with the motor cortex as well. So the muscles that are controlled by this part of the brain also become, well, I won't say weak, but sometimes they could be slow to respond or even paralyzed. And a really good example of this is a muscle in the back of your throat called the epiglottis. And what's actually a little flap of tissue, but it's controlled by a muscle. And if someone eats, that epiglottis closes the passage of the food so it doesn't go down into your trachea. It goes into your esophagus, so that it goes to your stomach and not to your lungs. But what happens during intoxication is if you were to vomit, the vomit will come up the esophagus, and then if that epiglottis is paralyzed, it can't close. So then the vomit will come back down into the trachea and into the bronchioles, and of course the gastric acid, that acid will basically destroy the lining of your lungs. And if it's bad enough, of course, you're dead within 24 hours. If it's not that bad, then instead you'll get what we call aspiration pneumonia. And that can be bad enough that it'll take about 3 or 4 days, and then you'll die. So either fast or slow, but you'll probably die. And the drugs are not usually good enough to prevent that, especially if it's a massive infection. It's very rapid growing. And of course, we hear about these stories all the time with the drinking games, that go on in colleges, and that's what happens. Okay, so what else happens here? Let's look at how this actually works. So biologists, of course, will remember the garden variety neuron. Maybe even the chemists in the group remember this when you guys were in high school. And you remember that the electrical impulse on a neuron travels from the cell body or the soma down the axon toward the terminal. And at the terminal, that electrical impulse releases a neurotransmitter. And back here on the cell body, those are the dendrites that have receptors on them. The receptors can also be on the cell body, and the receptors are the receivers of information. Okay? Now, I have a nice little diagram, a nice little movie that we're going to show. It's only a minute or two. And it shows how this actually happens. It's a movie that I made with a colleague, and you are all going to get copies of this movie. It's 3D animation. It's very cool, and I think you'll like it. But Myra's going to click it on now.
An electrical impulse moves down the axon toward the terminal. It signals the vesicles to release acetylcholine into the synaptic space. Now, once the acetylcholine is released from the nerve terminal, it binds to special proteins in the receiving cell called receptors. This binding of acetylcholine to its receptors causes a channel to open and sodium ions to move into the receiving cell. As these positive charges enter the cell, an electrical current flows across the membrane. When the neurotransmitter comes off the receptor, the channels close and the current is turned off.
Did you like that?
Yes.
Yeah, it's pretty cool.
Yes, it was very nice.
Pretty cool, yeah. It shows, this was for another a neurotransmitter called acetylcholine. Some of you may have heard of that. But it was just for illustration purposes. And what I'm going to tell you now is exactly the same thing, only with a different neurotransmitter system that alcohol interacts with. And so one of the neurotransmitter systems that alcohol interacts with and binds to its receptors, which, by the way, look exactly like the ones you just saw in the movie, is called glutamate. Has anyone heard of glutamate?
Yes, we've used the National Institutes of Health website that has a neuron lesson in it that the kids have an interactive they do with glutamate and the other one as well.
Oh, wonderful. Now, who's telling me this? Because I couldn't see who was talking.
Gail Wortman and Tamwa.
Oh, great.
Iowa.
Great, Gail. So, yes, the NIH has one of those series that talks about alcohol. And the glutamate receptors, I know others of you have heard of glutamate because I think you've probably all had Chinese food that has monosodium glutamate in it, right? Same glutamate. It's the same thing. It just has one sodium molecule associated with it to make the basic salt, right? Add a little...
I thought maybe you were going to tell me that if I ate Chinese food, I would have better neuron reception.
Well,
Causes a headache?
it is what causes the headache. It is what causes the headache.
dear.
You've probably got enough glutamate there to keep your memory going just fine and your cognition. So not only glutamate is one of the targets, but also another neurotransmitter called GABA. These are both amino acids, by the way, and the receptors for both of these neurotransmitters look just like this cartoon, and they work the same way as you saw in the little movie. But let's look at this a little bit more closely, how alcohol interacts with these receptors. So it turns out that alcohol actually depresses the firing of these neurons. So normally they fire, but in the presence of alcohol, it slows them down. Okay? And this is what's responsible for all the effects of alcohol that we know. And we're going to look at these receptors sitting back here on these neurons to make this happen. So in the case of glutamate, glutamate normally increases the firing rate of a neuron. Normally it increases it. So if alcohol is going to decrease the firing rate, it must block the effect of glutamate. Does everybody see that logic?
Yes.
Okay, so alcohol blocks the glutamate receptors, and that is responsible for affecting your judgment, reducing your memory, a little bit of memory problems, a little bit of cognitive problems. Okay? And on the flip side, if we look at the GABA side of the story, GABA does the opposite of glutamate. GABA decreases the firing rate of neurons normally, and what alcohol does is it boosts that effect. It makes it even more. So alcohol actually holds the GABA receptors, those channels that you saw in the movie, holds them open for longer. They can't close. And that lets more negative charges in the cell. And I'm going to show you that in a picture in the next slide. But what's responsible for that, what that's responsible for are some of these other aspects of alcohol's actions. The incoordination, the reduced memory, slurred speech, the disinhibition that we hear, that we all see, you know, the obnoxious part of the alcohol. That's due to your actions at GABA receptors. And here's a little cartoon of a GABA receptor. And the one in the curriculum is a little bit different from this because it's animated, but it shows the same thing. And this just shows that there are chloride ions this time, not sodium like in the movie, but chloride. And they go through this channel, and that channel is held open longer when alcohol interacts with this receptor. Okay? And the more negative charges inside the cell makes the cell turn off its firing rate. It inhibits the firing rate of those neurons. All right, so let's take one example of what happens in the brain. There is a part of the brain called the hippocampus I mentioned before that's responsible for learning and memory. And alcohol causes disruption of hippocampal function, and that is what underlies what we call the blackout. So the blackout is when someone cannot recall certain events that may have happened the night before while they were drinking. And that can be very mild. Maybe they forgot someone's name, they forgot a few things, or it can be very serious because they can't recall an entire event. And that can be with as little as 2 drinks, especially in an adolescent. The sensitivity of the adolescent brain is very, very different than the adult brain. They are more sensitive to the memory loss and the damage to alcohol, but they are less sensitive to the intoxicating effects, meaning the sedation and the motor incoordination. So it's kind of a, when you think about it, a cruel fact of life. It's kind of like, oh, I can drink more, but in the process I'm going to do more damage. And that research has just come out in the last couple of years. It's something very new. We didn't know this 10 years ago. And actually, with the help of MRIs, magnetic resonance imaging pictures, these pictures are taken from 2 people. The person on this side right here is a normal person, and the person on this side right here is a person who began drinking as an adolescent. This is a person with what we call adolescent onset alcohol use disorder. long term there. A person who started drinking as an adolescent. Oops, didn't mean to do that. And these are MRI images. They're taken this way. If you look at me for a second, they're taken this way through the brain. Okay, so you're looking this way. And so you're looking at the hippocampus. Right here, I've circled it in red for you. In the normal adolescent, it's about that big. You can see kind of in the middle. I'm sorry, your slides are very small, so it's probably hard to see. But that's normal. And then in the adolescent who started drinking, you can probably see that the hippocampus is smaller. Can you see it? Maybe my finger is blocking it. But there it is, right there. And on the here you can really see the difference. You see that? Well, so what? So they have a smaller hippocampus. Well, that's not so good. Turns out, at least the most recent research tells us that the smaller the hippocampus is as you start to age, the more likelihood that you will get Alzheimer's disease. And so this is a really good example of maybe you might not be genetically predisposed to getting Alzheimer's disease. Maybe your parents didn't have Alzheimer's disease, but because of something you are doing yourself, your environment, you are increasing your own risk. It's not necessarily what your genetics have dealt you. It's something that you are doing yourself. And that's really new information. That just came out in the past year. So what can explain this shrinkage of the hippocampus due to alcohol? Well, it turns out that from animal studies we know that in fact, actually this is pretty new information as well. I'll bet you guys didn't know that even today, as you are sitting there, that you have new neurons being born in certain parts of your brain. Did you guys know that? You know, most of us were taught that neurons were not born after we are born, that we have everything we have when we're born. And that's not true. We know a lot more now. And the hippocampus is one of those areas that has ongoing neurogenesis, birth of new neurons. And it turns out that there are stem cells there that give rise to new neurons. And look at the difference between animals in this case that have, these are the control animals and the drinking animals. And by the way, this is after a single binge. After a single binge, there's about a 40% decrease in the stem cell proliferation, and a month later, you can see more than a 50% decrease in their neurogenesis. And this can help explain the memory problems that we see in adolescents that, and as well as adults, that occur with alcohol use. And when I say use, I'm not
Is
talking...
that reversible?
Great question. We want you guys to ask the questions we think your kids are going to ask you. And that is one I think they're going to ask. Is it reversible? So yes, actually some recent studies have shown that this is reversible. What we don't know yet is whether the neurons that are coming back are normal. So whether now there's some damage to the stem cells, whether they give the same kinds of neurons that will make the same kinds of connections that they should make. That we don't know yet. Okay?
Do they know how?
Hi, go ahead.
Do they know how long it takes for that regeneration to occur, and if there's anything that can be done to assist in the process?
No, we're not there. We're not there yet. These are from animal models, and this is very, very new research. But everything that we've shown in the animals, we know now so far happens in humans. So we're pretty excited about finding this, and I think in the next 5 years you'll hear a lot more about it. Whoops.
Is it, question,
Yeah.
is this, as I look at this slide with alcohol decreased neurogenesis, is that going to be additive then? In other words, after one binge, we have this pretty marked decrease. If we binge twice, will we have a linear decrease, or will it be pretty well leveled off, or what?
Well, we don't know that, but I can assure you that we don't lose all of our neurons in our hippocampus. You know, the picture that I showed you with the MRI did show considerable shrinkage. I do not believe there are any studies in humans that show that the hippocampus is, you know, more than 75% reduced. I think there is a certain limit, and certainly in alcoholics who, of course, have gone through 30 or 40 years of drinking, they have considerable other changes in their brain, including a shrinkage of the hippocampus. But I do believe that the hippocampus is one of the first places where you see this happen. Okay? Unfortunately, we can't answer the question with that kind of precision yet. All right, so what can explain this decrease in neurogenesis and stem cell proliferation? Well, it turns out that these cells are being killed off by a mechanism called apoptosis. How many of you biologists in the group teach about cell death? Anybody?
I do.
Oh, good. Do you talk about apoptosis?
Yes.
Oh, wonderful. Okay, so we're going to talk about this a little bit more in the next week, and this is a great example of discussion about how cells undergo cell suicide, or apoptosis, a genetically programmed event, and this occurs during alcohol binging. So this does explain the decrease in the percentage of alcohol is in a beer. Sometimes it's 3.5%, sometimes it's 5%. But in a typical 12-ounce glass of can of beer, I can't remember if it's 4%, 3.5%. A glass of wine, a 4-ounce glass of wine, or a shot of hard liquor is all the same amount of alcohol, same thing. A drink is a drink given those definitions. They all have the same amount of alcohol, and this is a fabulous opportunity for your students to figure it out. We have activities that we want you guys to make, and we have put some in the curricula as well that deal with math. And this is a wonderful opportunity for the kids to figure out the alcohol concentrations based on a drink and what the definition of a drink is. There is a lot of mythology going on that one beer is not the same as one glass of wine. In terms of the alcohol, it absolutely is.
been my understanding that the liver can process about an ounce an hour. So if you drank an ounce an hour, your body could keep up with detoxification. Is there any truth to that statement?
Well, that's something that the bartenders like to use as a rule of thumb so they don't get sued for serving people when they walk out the door. And Suzanne is going to explain to you how that sort of rule of thumb doesn't hold true for everyone, and maybe a few people out there, but I'll tell you, I have been an expert witness in several lawsuits where people believe that, and they certainly show up quite drunk on the road. So we're going to discuss that after your potty break.
Okay.
Great introduction into that second part. So we're going to keep this very strict. Please keep your microphones on because we're going to talk as you come back, and 5 minutes exactly. Thank you.
Great. So we just spent some time with Shelley talking about how ethanol can get in and around the body. And now we're going to spend a few minutes talking about how the actions of ethanol are terminated in the body. And I'd like to start with a common misconception. In fact, one of the most common misconceptions about alcohol is that it leaves the body unchanged in the urine. This actually isn't true. In fact, most alcohol is broken down by the body and eliminated in a process known as metabolism that Shelley touched on a bit earlier. Now, if we go back to our diagram of the circulation, unless you're inhaling ethanol through one of the vaporizers that is now out on the market that I think Shelley mentioned, ethanol coming in by ingestion has to go through the stomach, and we saw that earlier. And as it moves through the stomach, a little bit of alcohol is actually metabolized, and this is a point that we'll come back to in just a minute. And then what we know actually is that most alcohol survives the stomach and enters through the bloodstream, is absorbed in the small intestines where there's a lot of surface area for absorption to happen. Now from the small intestines, alcohol can travel in the venous blood to the liver. And it's here in the liver that most of alcohol metabolism takes place. Any alcohol that survives the liver then moves back into the venous circulation and can be exhaled in a small portion in the lungs. Now, any ethanol that makes it through the stomach and the liver and the lungs in that first pass continues to circulate throughout the body in the bloodstream and continues to be metabolized or exhaled as it circulates through these organs until it's completely removed. So now let's consider each of those steps separately. Okay, so here we are in the stomach. What is this term first pass effect? Again, we pharmacologists use this term first pass to describe the amount of ingested ethanol that gets metabolized on its first pass through the stomach and the liver. Okay, so as I mentioned earlier, only a small amount of metabolism of ethanol occurs in the stomach. And this amount is actually reduced even more if you're a female. And that's because females actually lack the enzyme that does the metabolizing of ethanol in the stomach. What about the liver? There's also a first pass effect metabolism of ethanol in the liver. So in the liver, unlike the stomach, is very efficient at metabolizing ethanol. In fact, the liver is an organ that's chock full of enzymes and is responsible for doing a lot of detoxification in the body of other drugs that might enter our bloodstream. Now, as I mentioned, the liver is very efficient at this job, but compared to males, females again do a very poor job of metabolizing alcohol in the liver. So taken together with the differences in gender body composition that Shelley touched on earlier, you can see these are these two differences in metabolism of gender gender differences in the metabolism of ethanol contribute to a difference in the ability of a female or a male to be intoxicated. So females are more likely to become intoxicated from the same drink than a male if you put those three ideas together, the body composition and the metabolism in the stomach and then in the liver. So what about metabolism? Let's consider it at a cellular level. Okay, so ethanol is actually metabolized ultimately to acetic acid and water. And here we are in the liver. Let's pretend again that we're the ethanol molecule, and we've just entered the liver through the portal vein. We can diffuse across the cellular membrane here into the hepatocyte. And now we're in the cytoplasm of the hepatocyte. Here ethanol encounters ADH, the enzyme that metabolizes ethanol to its intermediate acetaldehyde. Now, men are much more efficient at doing this metabolism from ethanol to acetaldehyde because the enzyme ADH in men is much more active than the enzyme in women, again contributing to the fact that metabolism of ethanol happens more efficiently in men than women.
Can you repeat that?
So this enzyme ADH in men is much more efficient at doing its job than the same enzyme in females.
Okay.
So females can actually carry out this process. They have this ADH in their liver, but actually it just happens less efficiently. Men are able to do this more efficiently. Is that clear?
Yeah, but is the ADH different in females?
Oh, we're going to talk about
just
that
more
actually.
active?
We're going to talk about
Okay.
that in just a minute. So,
Thanks.
sure, no problem. So acetaldehyde then in the mitochondria can be converted to acetic acid and water, a byproduct of this reaction. And that happens when acetaldehyde encounters the enzyme aldehyde dehydrogenase found in the mitochondria. Now, ethanol metabolism for you chemistry teachers actually happens, both of these reactions are oxidation reactions. And in this case, the enzymes are taking the electrons away from the ethanol to produce acetaldehyde. And we're going to look at that in a little bit more detail at the molecular level in the next slide. But first, I want to remember to tell you that this intermediate acetaldehyde is actually a molecule that you're probably familiar with, just maybe not by name. So acetaldehyde turns out to be a very toxic intermediate. The process of metabolism is actually in place to terminate the actions of ethanol in the body, but acetaldehyde turns out to be bioactive in that it's the molecule that is responsible for all the classic symptoms of a hangover that you might associate. Sweating, rapid heart rate, nausea, dizziness, the headache that's so commonly associated with a hangover, and at the cellular level it actually poisons the cells. So you can see that acetaldehyde buildup wouldn't actually be a good thing. And that's why the process is actually two steps. And the acetaldehyde is then further converted to acetic acid in the mitochondria. Okay. So just to review again, the two-step oxidation process, ethanol is converted to acetaldehyde by alcohol dehydrogenase. And then acetaldehyde is converted to acetic acid and water by acetaldehyde or aldehyde dehydrogenase. Now let's look at this enzyme-substrate relationship in a bit more detail. Where ethanol is the substrate.
I'm sorry, I have a question.
Yes, sure.
I have a question.
Let's go back.
This acetic acid, then, it's deposited in the blood. Is that
I believe the acetic acid then is resorbed into the body and enters the Krebs cycle and becomes part of the natural process of the metabolic pathways in the body.
well, what I was leading to is this what can cause acidosis?
You know, I'm not sure about that. Shelley, do you know? if that's the bottom line for acidosis.
Oh, one gets bigger.
We've got a lot of discussion amongst everyone about whether this is true or not.
I do know that as the liver starts to become diseased, and the actions of alcohol in disrupting normal cellular metabolism, there's a lot of ketosis that can occur. In terms of acidosis, I can't answer that question. That's something we can maybe talk about
Yeah, we
next
can
week.
look that up.
Because I'm sure one of
a
your students
distant,
might ask that.
A distant reference to that would be the movie or book and drama.
So let's go on to look at the enzyme-substrate relationship. Now, for those of you chemistry teachers, this is probably going to look familiar. This, to orient you, this is an individual ethanol molecule, and Shelley sort of talked about this molecule before in more of a space-filling diagram. And along the outside here, we have the active site or the docking site of the enzyme ADH. And these red groups here are actually reactive groups on the ADH enzyme. So we've highlighted them here to show you how they interact with the ethanol molecule. So ethanol is docked at this active site in the ADH enzyme. And it's held there in place by two types of bonding forces. The first is a relatively weak form of van der Waals forces called London forces that interact with the hydrophobic end of the ethanol molecule. Okay, and on the other end we have interactions with reactive groups that I mentioned, hydroxyl amino groups here on the ADH molecule, and the polar end of the ethanol molecule. Normally this oxygen is very electronegative, and it, as Shelley had said earlier, it likes to pull the electrons away from neighboring atoms. And in doing so, it leaves the neighboring atoms with partial positive charges. And these neighboring atoms actually compete, or the ADH reactive groups actually compete with the oxygen for these hydrogens and their electrons. In fact, stabilizing the ethanol molecule in this binding pocket and making it energetically favorable for this oxidation reaction to occur. And if we look at the next slide, what we'll see is that ADH wins. It actually is able to scavenge these 2 electrons and 2 hydrogen atoms from, so it loses 2 electrons with those hydrogen atoms. Okay, so we've been looking a lot at the cellular and now the molecular level, so we're way down at the atomic level here talking. But I think the students are going to be interested in what does this mean for me. I'm a person, I'm an individual. So let's zoom back out and consider why it is that each of us or groups of us respond differently to alcohol in our bodies. And as it turns out, and this is going to touch a bit on where Shelley left us off in the discussion about metabolizing alcohol, we actually have 7 different proteins that code for the ADH enzyme, or the alcohol dehydrogenase enzyme. And all of these participate in some way in metabolizing alcohol. ALDH, or aldehyde dehydrogenase, there are 2 major proteins that participate in the metabolism of alcohol, and we have both of those proteins as well. So it turns out that each of these 7 proteins in the ADH, if we consider ADH for a minute, is coded for by a separate gene. So 7 proteins, 7 genes. Now let's talk about ADH7 first. ADH7 is actually the form of the alcohol dehydrogenase that's found in the stomach. And every one of us in the population seems to have the same gene coding for the ADH7 in our stomach. So we call this gene monomorphic. In other words, it only comes in one flavor. Now, I said earlier that women seem not to have ADH7 expressed in their stomach, while men do. So everyone has this gene, women and men, but the expression of these genes as proteins is not uniform throughout all the tissues in the body. In fact, again, we see that gender differences in the expression of this protein, ADH7, although both men and women have the gene. So there's diversity both at the protein level and at the level of the gene. Now let's consider ADH2. Again, another enzyme that catalyzes the reaction of ethanol, metabolism of ethanol to acetaldehyde. ADH2, unlike ADH7, comes in 3 flavors. Now, the scientists are so creative that we've come up with wonderful names for these enzymes. They're ADH 2.1, 2.2, and 2.3. Very creative, right?
I can follow that.
So why should we care about this? Well, I guess the first point I want to make here is that because there's more than one flavor of this gene expressed in the general population, we call this gene ADH2 polymorphic. All right. So in contrast to the monomorphic gene ADH7. Now why is that important? Well, as it turns out, most Caucasians, for example, inherit ADH 2.1. While most Asians inherit the gene ADH 2.2. Turns out that ADH 2.1 is slow at doing its job. It's not very efficient at metabolizing ethanol to acetaldehyde. Now, ADH 2.2 is actually very good at metabolizing ethanol to acetaldehyde, so individuals who inherit the ADH 2.2 gene actually turn out to have an increased buildup of acetaldehyde in their bodies. And at first you'll say, well, that's not a good thing because acetaldehyde is very toxic. But as it turns out, this is a strong deterrent to continue drinking. And what we find in the literature is that the Asian population has a decreased risk for alcoholism, probably in part because of this deterrent or this buildup of acetaldehyde that you can trace back to their genetic inheritance, ADH 2.2 versus an ADH 2.1 gene in Caucasians. And again, all of these processes will be affected by the next downstream metabolism that changes acetaldehyde into acetic acid, which is ALDH, and has its own story about how it's different in different groups or populations in the
Well, that's why ours went weird.
Okay, so any questions about this? So at this point, I think
Are there any other genetic predispositions on one or two, or is it mainly just Caucasians and Asians?
There are actually in the literature, and part of our resources will in the modules that are part of the curriculum will point you to different places to find the information for all different types of ethnic groups and profiles. For example, there's some literature that suggests that African Americans that inherit ADH 2.2 and 2.3 have an increased risk of fetal alcohol syndrome if they're drinking while pregnant, for example. I think there's additional information for American Indians as well as other ethnic groups, depending on what we know about the genetics today. So I think this is an ongoing research, and new information is coming out all the time about what combinations predispose you to an increased risk of alcoholism. So at this point, I sort of want to diverge a bit from the PowerPoint to go back and answer a question that came up for Shelley, and I think that was someone suggested that if you can metabolize drinks at approximately 1 hour, can't you account for that time frame and adjust your drinking in a way in which you would not become intoxicated? And actually, at this point, what I'd like to tell you is that these ADH proteins as enzymes are limited in our body. So we don't have an unlimited source of them. And what happens is they get saturated as we begin to drink, and the alcohol interacts with the enzyme, and the enzyme starts to do its job. And as it becomes saturated, adding another drink just puts you a little farther behind every time. And what ends up happening is these enzymes become overwhelmed in number by the ethanol molecules, and are not able to do their job as efficiently. So you become farther and farther behind to the point that you actually are impinging on increased intoxication, simply as a result of overwhelming the enzymes. Does that answer the question that came up earlier? about whether
It does. It answers my question. It's still not a bad policy because by then you hope the party's over.
well, certainly anything that would slow down the drinking would probably be helpful, including having food on board, etc. But I guess our mindset was that we didn't want to promote a policy that from the outset. All right, so let's see, where are we now? So that's basically the story of metabolism, and I did say that as the ethanol molecules pass through the stomach and the liver, where most of this metabolism takes place, some of it will still escape into the venous blood and can be exhaled in the lungs as a vapor. And we know that this is the basis of the breathalyzer test. And I want to look now at the anatomy of the lungs and their function and how they might facilitate this vaporization of alcohol. Now, most of us are familiar with the anatomy of the lungs, especially you biology teachers. I'm sure you teach this in class, that they terminate in these grape-like clusters of alveolar sacs. And each of us has millions of these alveolar sacs. Every one of those sacs is wrapped in, and these sacs are very thin-walled. And each one of those sacs is wrapped in a capillary bed, and you see that here in the picture. So we have both deoxygenated and oxygenated blood coming in through the capillaries, and each one of these is wrapped around the alveolar sac. In the small blow-up picture here, we see the blood vessel running under the alveolar sac. And what we want, okay, so here we are in the alveolar sac in the lungs, just one of millions of alveolar sacs that we have in the lungs. Ethanol dissolved in the blood can diffuse into these alveolar sacs. And inside the alveolar sac, there's actually a very thin liquid or mucosal layer that lines the inside of these sacs. And there, dissolved ethanol lives basically until it vaporizes into the airspace above in the alveolar sac. When it does so, this vapor can then leave the body during exhalation, and that's the basis of the breathalyzer test. Now, we know that because this ethanol that's vaporized in the alveolar sac is actually in equilibrium with the ethanol that's dissolved in the blood, we can get an accurate estimate of the BAC, or the blood alcohol concentration, at the time of the breathalyzer test. So what makes ethanol a volatile molecule? How about some of the chemistry teachers in the group? Anyone want to take a stab at this? What are some important properties of ethanol that make it a volatile molecule?
Only half of it's got the hydrogen bond.
Yeah, well, that's good. You weren't peeking, were you? That's okay. So anyway, ethanol compared to water is set up a little differently in that it can make about a maximum of 3 hydrogen bonds with neighboring molecules, while water, also a molecule that's dissolvable, right? These hydrogen bonds can be up to 4. And less hydrogen bonds means less bonds to break when vaporization is going to happen and release the liquid into a gaseous state. And another good example of this would be if you were on the other side of the room and someone opened their Kool-Aid container, you would be sort of hard-pressed to notice because there's no smell associated unless you happen to see them. But if they were on the other side of the room and they opened a jar of ammonia, I bet you would notice. Ammonia is a molecule that looks a lot like water, only it can only make one of those hydrogen bonds. So it's even more volatile than ethanol. So again, the structure of a molecule contributes a lot to the way in which it vaporizes, and this is one contributing factor to the volatility of ethanol. So at this point, I think I've told you just about everything that I can tell you at the moment about metabolism and elimination of alcohol in the breath. And so I think at this point, what I'd like to know is, Does anybody out there think that there's a reason why you might want to teach alcohol using basic biology and chemistry concepts? Does anybody see that this might be helpful at all in their classroom?
Well, it's
Can I ask
certainly
a
on the
question?
students' agenda.
Yeah, I think it is on their agenda, and that's one of the reasons why we chose this as a topic. It's certainly a topic that they are going to hear a lot about, and that
There's this drink called Everclear that's very, very high proof.
yes,
And the discussion with kids is that if you drink that, you will get intoxicated much faster. Is that simply because of the proof, or is that able to be absorbed more rapidly in the stomach, or what? Is it just the fact there's so much alcohol?
I guess I would say that higher proof means a higher percentage alcohol per drink compared with beer, for example. And so that,
Way higher.
yeah, so that in and of itself means that you're dealing with a higher concentration of alcohol from the very beginning. Yes.
But
I
the
think it's
absorption,
twice.
the absorption path though is exactly the same.
Oh yes. Still
Got
the
it.
same
Thanks.
ethanol molecules. Yeah.
Great, thanks.
I
Glad,
think it's
I'm
like
glad to see that
200
you have your
proof,
pulse
isn't it?
on what's going on.
There's another question.
Do we have another question?
I was just saying, I think Everclear is like 200 proof as compared to the usual 100 proof. So I think its percentage is like doubled.
Yeah.
200 proof is 100% ethanol.
Right.
I think Everclear is about 95%.
Yes. Not quite right. Yeah.
100 proof is not 100%.
Yeah, the consensus here is that it's somewhere in the 90s, high 90s.
Right.
And that's another example
Percent.
in teaching chemistry. It's really, really, really hard to distill something and get it 100%. So the Everclear is usually like 95, 96%.
And by the way, in the modules we cover this. In researching this, we found out that the origin of the word proof goes back to a time when you had to prove by being able to light by fire the alcohol, and whether or not it lit and burned with the correct color flame was proof of whether or not it was a certain percentage alcohol.
The other thing that you're talking about when you say why teach about alcohol, this is such a good argument for interdisciplinary information, and it makes you wonder why we ever split up biology, chemistry, and physics to begin with.
Yeah, it would be great in an ideal world, you're right, if we could teach these together.
Well, historically, we didn't know all of the biochemistry. A lot of the things we're only learned in the last few years, but now I don't know how one can study biology without knowing all this chemistry.
Correct.
Well, I know that back in the early part of the century, they decided that it would be biology and then chemistry and then physics in an order in high schools because they chose to go alphabetically.